Bonjour everyone. So the title of my presentation is Taking Neuroengineering Research from Publications to People. First of all, I would like to focus over here. The subtitle is From Publications to People. What does it mean? So this is a very popular author who writes articles and develops curriculum at Johns Hopkins University. According to her, that most of the PhD students nowadays are being trained in some specific domains, but they are mostly following what the supervisor is telling, and at a later stage when they go out of academia, they're not able to do some kind of independent thinking in terms of coming up of novel ideas and putting their research into applications which is solving some real world problems. If you go further into this topic, where this problem is coming from, a lot of times in academia we know that's the known thing, we focus a lot on publishing or otherwise we will perish. And this is not just limited to students, it's also applicable to professors, even people with permanent degrees. For promotions, we have to focus on publication a lot. And if we manage to publish in a good journal, we still have to keep publishing in a higher impact factor journals, otherwise we'll perish. So of course, it's quite evident that there is a gap between academia and industry, which is often talked about, and that's why a lot of good students also leave academia to join industries. So we need to bridge this gap. And to look at a little bit more recent example, this was an article published in MIT Press that hundreds of AI tools have been developed to catch the COVID, none of them work. Well, according to me, maybe none of them work would be an overstatement, but yeah, it's quite true. There was a surge in publications and most of them didn't work. So there's another tutorial which is made by the Google Research product manager, and she's a physician and doctor specializing in product development. According to her, there are 3 common myths associated with building models in healthcare, and that's why we see a surge in publications, but they don't necessarily translate, or the translation to the actual product solving the real world application is quite slower. She says that the main reason could be that more data is all you need for a better model. That's one of the myths. And another one is that an accurate model is all you need to make a useful product. So more data and accuracy. There is another thing related to even if you manage to cover both of these together, a good product is not sufficient. So in this presentation, with the time limitation, I'll be focusing on the first 2 aspects, giving my point of view related to electrophysiological research to be more specific. And since audience is heterogeneous, I'll be talking about what is neuroengineering, my research interest, and I work on behavioral neuroimaging and electrophysiology. And the myth I will be busting over here in the electrophysiology part. So what is neuroengineering? And it's actually used synonymously with neural engineering, and thanks to Mr. Elon Musk, it's also used in synonym with the neurotechnology, what the Neuralink company is about. According to a definition provided in the journal Springer, it is a biomedical engineering field, and that uses engineering techniques to understand, repair, replace, or enhance neural systems. And it's you can see it's a very interdisciplinary field, and it's also very evolving. And we and my contribution in the group, I work on behavioral electrophysiology and neuroimaging stuff, and integration of all these 3 sometimes can also lead to some kind of assistive technology development. In this presentation over here, I will be giving snippets of behavioral and neuroimaging studies, starting with that, and finally I will be ending with electrophysiological application. Won't be talking about assistive technology, that means like prosthesis or brain-computer interface and things like that, not today. So, and to be more specific, I have taken some examples of related to epilepsy over here, the tool which we developed, and also the processing part related to big data analysis on rodents for neuroimaging part. Starting with behavioral studies, so this was a work done in collaboration with the MIT and in India, where I did my PhD from, related to the developmental visual perception and cognition of studying these things of people who had cataract in the beginning, and then they were operated and gained vision. So what the hypothesis of all these studies was that based upon the Nobel Prize winners Human and Tossell in 1981, they had the studies on kittens where they were devoid of vision in the initial phase of their life. And they saw that it cannot regain vision even if it is operated or the patch is removed later. So they extrapolated this studies onto humans, and because of that, unfortunately, thousands or millions of people worldwide were devoid of vision. So the group actually challenged this hypothesis, and they were successful to show that they can gain vision. So actually it opened up an area of cognitive science where you can actually see the trajectory of vision being developed inside the brain. So the main open question, what they were trying to ask her, can brain acquire visual perception, cognition skills late in life, or is it learning which is just related to critical period? And based on this observation, there were a lot of behavioral and multimodal tasks being developed. And this is just an example of a lot of studies which long-term transversal research led to a large impact in people, of course curing a lot of people who were congenitally blind. And this was the first study in the world actually. The second part is this, the neuroimaging studies, which was my recent work done in Denmark in collaboration with the IRB Barcelona. We are trying to push the limits of 3D image processing here. So I believe most of you in the audience might be familiar of that MRI is used in humans to get the images of the brain. But are you also familiar that it can also be used to create this kind of beautiful map, which is basically the connections inside the brain, how it is being connected from one area to another, where different colors actually show the different direction of the fibers being connected. And this is very critical in studying diseases like Alzheimer's disease, epilepsy, stroke, cardiovascular, just to name a few. So cardiovascular listed because it's also used for fiber tracking in heart as well. So, but it is always very important that for a drug to come into the market, it has to be undergone some kind of preclinical trials, which is not humans. So these are the smaller scanners, but still powerful, like 7 or 8 Tesla. And you repeat basically the same process. This is used for drug discovery, treatments, and making more brain cognition discoveries. The initial part actually requires you to put the cleared brain, which is there in my pocket. If you want to see, you can later see. This is how crystalline it looks like, and then you image it. And firstly, comparing the existing technology, which is MRI, which was already much more frequently used for this kind of purpose, we see that if we zoom into the MRI, it gets pixelated. Then that means obviously the resolution is very important. If we are tracking the fiber connections, we need to have a clear resolution where the fibers are going from. So the crux is that the MRI resolution is in millimeters, whereas the resolution of the microscopes is in micrometers. And now this is our results from our study. You can clearly see visually how high resolution it is. It's actually 40,000 times better. Now people have been doing this kind of work using the chemical, by injecting chemicals inside the brain. And sometimes the chemicals can be one color per chemical also. But anyways, it requires a lot of money to put in using those chemicals, and there is a lot of cumbersome process which needs to be followed. The group actually developed a technique which doesn't require any kind of chemicals, and you can get the same or even better results tracking the brain fibers. And also you can do a big data analysis and get a 3D reconstruction of map like this. This is just a small area because the data is so in terabytes that it will not fit. A small brain produces a lot of high resolution data. So the essential novelties in the study are related to the new hardware that we developed, and also software which is related to the image processing, and also the later part which is related to tractography. But the main objective which was there, it doesn't require any kind of dye or tracer to do this kind of tracking. And you can actually do it in the entire brain. What I mean is entire brain, the conventionally used microscope, you need to slice the brain. If you imagine this has to be a brain, you can directly put it into the microscope and scan it. It produces a 3D image. So the last part of my presentation is related to epilepsy research, where I'll be busting the myth. So epilepsy is very well known, one of the most common diseases worldwide, and many people are affected by it. Yes, Napoleon Bonaparte used to also affect by it. And if I show you the picture, just whatever experience you have, out of these two signals, it's very difficult to check which one is epilepsy or not. So I actually gave this task to my students last week, and this was their response, the usual response, that the right one is epilepsy. But actually it's not. Both of them are collected from healthy individuals. It's just that one has, although same scaling, it has noise. That's it. Although the basic filtering was also even applied that the person moved. So EEG data set, considering it is very highly non-stationary in nature, it is also one of the largest biomedical data signals what we have. And even if I now show you the epileptic seizures, it's still difficult to characterize which one is epilepsy and not epilepsy, right? So just adding to the problem, the disease is also heterogeneous in nature, which means the heterogeneity not only varies across different seizure patterns, it's also heterogeneity is also there within the same patient. One day he will have different kind of seizure, the next day he goes to the clinic, he might have different kind of seizures. So even the patient-specific data systems are not that accurate. So obviously it's giving a hard time to your computer and also the clinicians. So can this process be automated? The answer is yes. And I already said that epilepsy is a very worldwide disease. The main thing to be observed over here, a ray of hope is that 70% of the cases are treatable if it is detected early. So just talking about more the heterogeneity and a little bit of myth related to these seizure signals is that it will always be in high amplitude range, which is not completely true. A lot of the seizure types has low amplitude, which is very difficult to detect for clinicians. And they even have a disagreement between themselves, that one clinician might say this belongs to a seizure, and other will say this doesn't belong to a seizure, it will be some other kind of activity which might look like a seizure. So they have even categorized formally categories like unknown onset, where they don't know where the seizure, how it is originating from, whether it is coming from both the hemisphere, single hemisphere, they don't know. And if they are not able to classify, they put it in the unclassified category to come back to it later. So this is actually very problemsome. You can imagine somebody crossing the road or driving a car, somebody will have seizure. So it's obviously very important to detect it as early as possible. But we are not predicting the seizures here. We are making the first step, that is the diagnosis phase. So the most common objective listed in the literature is that this is how the processing works, and let's assume that we have a signal which is very clean, and yes, you can clearly see the stages over here. What people usually do is that they segment the signals belonging to the seizure category and put it in a separate folder, which is called ictal. And also they do the same for the segments which are like over here colored blue in a separate folder, which is called non-ictal, and completely ignoring the electrode information over here. By electrode information, I mean if somebody has not seen the EEG, if you see this EEG cap, you put this kind of electrodes on top, the information over here is completely ignored. That means you don't know where it's coming from. So this is a common approach which is used for training the machine learning algorithms. So you basically do a binary classification between ictal and non-ictal stages. Ictal is seizure and non-ictal covers all the stages which is not epilepsy. In ideal world, this is how the signal should be coming from, but you obviously have a lot of noise, and this is how it looks. But assume you have done a very good filtering, you have everything covered, then you do a binary classification. So when I was talking about different kind of stages, there are interictal stages from the patient when it's not seizure, but still epileptiform patterns. And the epileptiform patterns is also pre-ictal, also post-ictal. Ictal is the only stage where it is seizure. Each of these classes have a different clinical significance, but here we are just focusing on binary classification for simplification. Okay, so now conventionally we apply biomedical signal processing algorithms and apply machine learning and pattern recognition, and voila, we get a very high accuracy. Now the problem solved. Well, not actually, the problem has not been solved. If you see closely over here, like what I told you in the beginning, the most common objective, people actually ignore the electrode information over here. And this is documented. It is called single channel EEG analysis in epilepsy. So they're not analyzing the continuous EEG, they're breaking down into segments and not using the electrode information. This is the most cited data sets in epilepsy domain. It was made by University of Bonn. I also used it a lot. And of course, there are many things good and bad about this data set. The most important thing which I want to just point it out is that when people do this kind of binary classification, where you have different stages against the ictal, people are completely ignoring that this data set also has the scalp EEG, which is from the scalp of healthy subjects, whereas the ictal stages and interictal stages were taken from the intracranial EEG. That means the scalp was cut open and you acquired the data on top. So this is actually what Elon Musk is mostly interested in, and also depth EEG. It's not covered here, but the noise level is quite less over here. It's quite noise free. So they are just classifying very different things as well. So of course, if you have more data, it also depends on what type of data they have. And in this case, not only is less data, but it's actually less annotated data. That's a very important thing people just completely ignore. So this is one of the myths. And if you look at the classification accuracy, what people have reached 10 years before, 100%, 99%, oh, super. But when we even look at some of the recent literature, people are making different combinations, are saying, and you got 99%, I got 100%, I'm better, and getting published in good journals. And even more recent. So what thing is changing over here is you're getting beautiful graphs, but nothing in terms of accuracy is adding value to the final product. So if you have a highly accurate system, it's not enough to make a final product. And this is the citations what this data set is talking about. I'm not saying it's a bad data set, it's just that people should have a well-defined problem statement, which is not correct for over here. So if you do a simple Google Scholar research, this is the number of citations or the hits, sorry, it gets 139,000. And if you, even if you be more specific related to automated diagnosis, so much of research is going on. And this is the number of citations this every year the data set is getting. I would define this kind of research as Newton came and discovered the law of gravity, and people are discussing that, okay, orange is falling on their head. Now this orange is also following the law of gravity. So this is a common kind of research going on. Our objectives was that to develop not only a coherent system which has high sensitivity and specificity measures, but finally a tool which can actually analyze the entire continuous EEG waveforms and help clinicians for better diagnosis. So basically we included the electrode information. This is a snapshot of the tool, and just to give you a quick demo of the tool, when you open it, the logo of my previous institute comes up, and what is required to be input is that only the sampling rate and the number of channels you need to analyze, and everything is then automated. So what it will do is this, you can see the waveform on the left and the forward and backward modeling over here. So what you are seeing over here is that whether the signal had epileptic seizures or not, and you are also checking whether how it was propagating on the right side. And you can actually save the session as well to just come back to where you started from. So just for the beautification, we also added, because radiologists are used to seeing grayscale values. That's it. Okay, so we are not saying we are the first one to do it. People have also done focusing on these kind of research before. A lot of the methods are patient specific, which means you need to train the data manually initially, and if the user is not trained enough to do the training properly, the entire algorithm doesn't work. And yeah, they are using different kind of measures. This was high frequency oscillations, and there is another measure which had good results, but well, the result sensitivity is quite varying, but anyway, it is using intracranial EEG again. And this is another model which actually used one of the similar feature what we used in our model, but they had patient specific. These are the people most likely we are competing against. They are using very high dense electrodes of 76, 96 channels, and also have MRI data. So in our study, we don't have the MRI data honestly to correlate if it was working properly. They have it anyways. Now where the research is going, this was a recent article published, Nature Reviews. And they say that basically the future is multimodal. That means you need to integrate these features with not only just one technology, with other type of sensor variable technologies to move ahead. That's what our inline research areas in Denmark. And we are also focusing on development of app which can provide a real-time EEG analysis to the users, and that can be deployed in the underdeveloped countries. So just to quickly conclude, it has high classification rate anyways. So the main features which we found out were importantly the energy, standard deviation, phase space, and root mean square values. And complex wavelet gave good results. It's completely automated. It's helpful for pre and post diagnosis. Telemedicine tool. It was also integrated with the EEG amplifier, which we developed, and we also developed a new repository. This was completed. This is ongoing. These are just the brief research highlights. Not only we developed the model, we also developed a data set which is publicly available, now well cited. People can use it to check a common grounds if their model is working properly or not. We also have a time series of data, and there is the tool can be deployed in clinics and working on. So yeah. So these are just rather related to epilepsy, which we are working on, prediction, that how well early you can predict where the seizure is coming. It can be 60 minutes, 20 minutes, anything. Using multi-objective optimization, more in the data science side, how to get the features and integration with the hardware. Using connectomics for patient-specific epilepsy. So the important myths debunked here is a lot of data sets will solve the purpose, not the thing. As you saw, higher accuracy is not at all the only measure which you need. Seizure detection is not a trivial problem. Many of the seizures might not be of high amplitude, and there is a different conferences going on just to detect seizures. And we are not replacing here humans. It is only to assist the clinicians to do a better diagnosis and quick diagnosis. I would just like to end that AI is a very powerful tool to find solutions, not the solution itself. Thank you.